Hello students, in this module we are going to be learning how an egg is formed in a female, the process called as oogenesis. Now when we talk about oogenesis or the formation of human egg, students, it begins almost 6 months before she is even born, and it completes only and only if a sperm fertilizes the egg. So let's see what are the various events which occur in the process of oogenesis. Now when we talk about oogenesis, students, we begin with the cells we call as the primordial cells. These are the cells of germinal epithelium of ovary, and they are diploid. That means having 46 chromosomes in the nucleus. These cells undergo rapid multiplication to form many cells which are called as the oogonia is the plural, and oogonium, students, is the singular, and the process by which this is happening is the mitosis, and this phase of rapid multiplication is called as multiplication phase. Now oogonium, students, undergo huge spurt of growth and forms a cell which we call as primary oocyte, and this phase of formation of primary oocyte is called as the growth phase because it is showing rapid growth. Now primary oocyte, students, you see, is also diploid in having 46 chromosomes in the nucleus. Now primary oocyte undergoes meiosis, and we know meiosis is meiosis 1 first and then meiosis 2. So it undergoes meiosis 1, students, to form 2 daughter cells which look like somewhat like this, and herein what you see here is, students, huge unequal distribution of cytoplasm. There is one big cell and then there is one very small cell, and these cells, students, the big one is called as the secondary oocyte, and you see the number is halved now. It is N. N means haploid set of chromosomes, single set of chromosome, because meiosis 1 is a reduction division itself, and the small cell which you see here, students, is called as the first polar body. But why is such an unequal distribution of cytoplasm taking place here? Because we'll see further, students, the secondary oocyte, the large cell, is going to further proceed to form zygote in embryo. So for the development of embryo, nutrition is required, and that nutrition is provided by the cytoplasm. So maximum cytoplasm has to be retained in the secondary oocyte and not in the polar body, students. From here in, students, this is called as the maturation phase. But now in a woman, students, further oogenesis will proceed only and only if a sperm fertilizes the egg. When a woman ovulates on 14th day, students, she ovulates at this stage of secondary oocyte, and only if a sperm fertilizes secondary oocyte, it is going to further proceed, and meiosis 2 proceeds, students, wherein again 2 daughter cells are being formed. One daughter cell, the big one, is called as the zygote, and the smaller cell, another very small body, is also formed, students. The big cell is called as the zygote or the fertilized ovum, which is diploid, and the small cell is called as the second polar body, because first polar body was already formed, and that first polar body, students, also by now divides further to form 2 more polar bodies. But these 3 polar bodies, students, very soon they all degenerate. So when polar bodies are degenerating, why were the polar bodies formed? See here, students, the first polar body which is formed, had it not been formed, secondary oocyte would have 2n number of chromosome, which wasn't right for fertilization. So to retain the haploid state of chromosome in the secondary oocyte, first polar body was to be formed. So this is, students, the process of oogenesis. In this module, students, we completed oogenesis. We are going to be continuing with understanding the structure of human sperm in the next module. Till then, take